Hello, this is our project on impact of combined sewer overflows on chlorophyll, nutrients, and phytoplankton at two sites near SUNY Maritime College. This project is by Eduardo, Argandona, Nadia, Gallardo, Victoria Nieto, and Katherine Schillat. So the objectives of this research is to compare chlorophyll, dissolved ammonia nitrogen, dissolved inorganic nitrogen, DIN, and dissolved inorganic phosphorus, DIP, concentrations at sampling sites HC and OR near SUNY Maritime, and analyze phytoplankton at site OR. So the two sites, HC, Hammond Creek, is a shore site down the street from Maritime College, and is on the eastern side of the peninsula where Maritime College sits. At site OR, it's a dock at SUNY Maritime's waterfront, and it's on the western side of the peninsula. The other objective is to determine whether combined sewer overflows, or CSOs, influence these parameters. So a little introduction and background. CSOs release untreated sewage and stormwater runoff into coastal water surrounding SUNY Maritime, specifically following rain events. Both combined sewer overflows and treated wastewater introduce dissolved nutrients, including ammonia and nitrogen, into the water. And this is according to O'Shea and Bronson. These nutrients are limiting to primary producers and can lead to algal blooms and eutrophication. So eutrophication is where there's an excess of nutrients in water, and this can deplete the water quality and the marine life by decreasing its oxygen levels. Ammonia, nitrogen, DIN, and DIP concentrations were measured to determine the influence of wastewater treatment on nutrient loading, and chlorophyll A concentrations were used as a measure of algal biomass. The plankton species were identified to determine if nutrients influence the community as a whole. The purpose of the overall research of field methods, ES451, is to determine if CSOs impact water quality at sites HC and OR, and if there is a difference in water quality between the two specific sites. The purpose of this experiment is to determine if combined sewage overflows impact chlorophyll and nutrient concentrations at both sites and the phytoplankton community at OR. We have a formulated hypothesis that CSOs will deplete water quality near SUNY Maritime, and that high nutrient levels will correspond with high chlorophyll concentrations and elevated phytoplankton. And here are the methods.
We collected surface water samples from sites HC and OR once per week for 10 weeks. Chlorophyll and nutrient samples were prepared by filtering water samples using a handheld pump and Wattman GFF filters. The filter paper was placed in a centrifuge tube and wrapped in foil for later chlorophyll analysis, while the filtrate was transferred to plastic bottles for later nutrient analysis. Chlorophyll was extracted from the filter paper using acetone and measured using the Turner flow meter. Ammonia was measured using procedures modified from the Chem-Metrics using the LabQuest colorimeter. Dissolved inorganic nitrogen and dissolved inorganic phosphorus were measured by the flow injection analysis colorimetrics using a latch head flow injection analysis machine. Phytoplankton samples were collected at site OR only using a plankton net. We analyzed the samples in the lab under the 10 magnitude objective lens of a compound microscope. Organisms were identified using the phytoplankton identification key. Less than 10 identified cells of a species was considered some, and more than 10 cells identified was considered elevated. Now on to results.
Our data for chlorophyll and nutrients are graphed over time. Figures 1A and 1B show concentrations of chlorophyll in green and ammonia nitrogen in gray over 7 weeks, which were the dates when we had data for both parameters at both sites. The stars indicate rain events, which we defined as sampling days when the 2-day average rainfall was greater than 0.1 inches. Comparing 1A and 1B, chlorophyll concentrations were greater overall at HC, where they range from 1.93 to 15.35 micrograms per liter. Then at OR, where they range from 1.68 to 7.05 micrograms per liter. Meanwhile, the average ammonia and nitrogen concentrations were similar at both sites, 0.23 milligrams per liter at OR and 0.345 milligrams per liter at HC. These values are above the EPA's 0.2 milligram per liter ammonia standard for polluted waters, but because our ammonia nitrogen concentrations include both ammonia and ammonium, we didn't make a direct comparison with this standard. Also, there were no clear trends between rainfall and chlorophyll or rainfall and ammonia, other than peaks in ammonia nitrogen at both sites on October 15th. These were followed by peaks in chlorophyll concentrations on October 22nd. In figures 2A and 2B, we plotted chlorophyll again in green, this time with DIN concentrations in orange and DIP concentrations in blue. As shown in 2A, chlorophyll at OR followed a similar pattern to DIN. For example, between October 8th and 29th, both chlorophyll and DIN decreased, increased, and decreased again. As shown in 2B, there was the opposite trend at HC, where increases in chlorophyll corresponded with decreases in DIN, and decreases in chlorophyll corresponded with increases in DIN. This was interesting because nitrogen is known to limit algal growth, which is measured by chlorophyll concentrations. It's expected that as DIN increases, chlorophyll will also increase, which wasn't the case at HC. These results, though, do compare to those from a study by Lee and others, where it was found that rain and tides resuspend particulates in water, blocking sunlight and actually reducing chlorophyll concentrations. Another possible explanation for this inverse relationship at HC is a lag in nutrient uptake by phytoplankton. For example, it rained on October 15th, and there was an increase in DIN from the previous week. However, an increase in chlorophyll was not measured until the next week on the 22nd. This brings up one of the limitations of weekly sampling. Rather than sampling daily, it's difficult to see whether there really is this lag between nutrient loading on rainy days and algal growth after the rain has stopped. There were no clear trends with DIP. One other thing we did with our chlorophyll, DIN, and DIP values was compare them to measurements from a site near the Maritime campus under the Throgs Neck Bridge that has data available from the New York City Department of Environmental Protection. We found the average chlorophyll, DIN, and orthophosphate, which can be compared to DIP, concentrations at the DEP site from September through November for the past 5 years. All of our average concentrations were similar to the 5-year averages, except for the average chlorophyll at OR, which was about half the average at the DEP site. We also did some statistical analyses to see if there were significant differences between chlorophyll and the 3 nutrients, and also between concentrations of the same parameters at the 2 sites. So we did one-way ANOVA tests, which just compare the means of, in this case, 2 variables to see if there's a statistically significant difference between them. So in other words, does one variable influence the other? And we found that there was a significant difference between DIN and chlorophyll, and also between ammonia and nitrogen and chlorophyll at both sites. And this indicates that nutrient loading from CSOs and wastewater in general may increase algal biomass and productivity. And we also performed t-tests.
While conducting t-tests, which are like these tests that used to determine the significant differences between 2 groups, in this case 2 sites, it was revealed that a statistical significance difference between the sites for chlorophyll, which gave us a p-value of 0.0381, and a DIN, which gave us a p-value of 0.00764, indicating that CSOs and treated wastewater may impact HC and OR differently. And because of that, that would imply that because the means are less than 0.05, it means that there's a significant statistical difference. And if we look over at figure 3, we can see that we have a graph of phytoplankton species at site OR, which this graph represents the 10 weeks that we took samples, and as shown, you can see how frequently elevated our plankton species were, and the most common was a continuous, which was a phytoplankton species at site OR, in comparison to Skeletoma, which was the most common species in the East River in the papers of Lieta and the Roshan papers, and this represented that there was no trend in rainfall and nutrients, and species elevations indicating that nutrient loading did not have a significant impact on the phytoplankton community. As stated previously, it is hypothesized that the CSOs will deplete water quality near SUNY Maritime, and a high nutrients level will correspond with high chlorophyll concentrations and elevated phytoplankton. And when we determine our test, our test stated that wastewater treatment in general depletes water quality surrounding maritime. Nutrient loading, particularly of nitrogen and in forms of nitrite, nitrate, and ammonia, is related to chlorophyll concentrations and may limit phytoplankton productivity. Due to this, more research with a greater number of samples is needed to determine if CSOs especially impact nutrient levels in phytoplankton. Due to this, we can't really see a decent trend, so possibly conducting a daily testing instead of a weekly testing could possibly give us a better trend. For more references on these type of subjects, you can look down at our work sites, and that concludes our presentation. Thank you for listening.
Thank you.